Well, good evening, and welcome to Bio 212, Chapter 25, Metabolism and Not So Much Nutrition. This is Chapter 25 in Tutor and Derrickson's Principles of Anatomy and Physiology, and you're listening to Bio 212 at Norwalk Community College. What we're going to do is we're going to focus on primarily what does the textbook mean when it thinks about metabolism? And what I want you to remember, of course, is that this chapter follows, this is your digestive system, right? Alimentary canal, stomach, small intestine, large intestine, and poop everything out. And remember, in blue, we had everything that was collected through digestion headed towards the liver. And that's the part your textbook fails to mention when they start all of this. So inside the back of your head, not just every single cell inside your body, but most of the processing we're talking about now is what's actually going on inside the liver. Especially when it comes to the regulation piece. You're going to see when they start talking about the effect of hormones on cells within the body. Mother Nature needs people to get materials that will allow us to make ATP, right? And even though your cells can make ATP, every single one of them can because they all have mitochondria. When we're breaking down materials that we've ingested, we are going to release energy. When we need to build things, which can happen, again, inside every single cell inside your body, we're going to take some of that energy and we're going to make more complex structures. Okay, so everything you ingested, regardless of what it is, hamburger, helper, makes a great meal, right? Lots of salt in it though. Okay. Send it to the liver. Liver will process what it needs, return materials back to your circulatory system, and then every single cell inside your body should have what it needs to make that ATP, along with everything else. Okay. Anabolic reactions don't just mean ATP. This is the energy that allows that to happen. You could be making amino acids together. Through dehydration synthesis, you can make proteins. Remember, they're the machines that actually keep everything running inside your body. But we need to fundamentally understand how we make the greatest number of ATP molecules inside the body. In the next slide inside your text, they throw this thing at you, okay, which is confusing as all get out. But what I want you to understand is that you can take simple molecules, just like I said, and you can create larger, more complex structures. The example, of course, is for glucose. The complex structure of glucose is glycogen. For amino acids, it's going to be proteins, and from fatty acids, we're going to build triglycerides. To build and store them requires energy. Okay, and in the process of doing that, you release heat. Likewise, when you break them down, the catabolic reactions, which is, of course, the breakdown part, is going to release heat as well. And then we just keep this cycle going for as long as you're alive. To transfer that energy, those two little arrows in the previous slide, we have oxidation reactions and we have reduction reactions. And big fancy way of thinking about this is you're going to remove electrons. And electrons are essentially the potential energy that is now being shifted from one location to another. We call it dehydrogenation. Big fancy word for saying you're taking away some hydrogens and you're going to transfer them. And you transfer them using nicotinamide adenine dinucleotide, and that's NAD, precursor structure for your B vitamins, and a flavin adenine dinucleotide, FAD. And anywhere you see these inside our lecture today, you're paying attention because ultimately they travel to one location and one location only. They are traveling to, in this class, the electron transport chain. And you'll see why that's important in a second. The example that's being used and introduced inside this class is that glucose is the primary energy source for the production of ATP. And remember, let's see if we can find a purple today so it makes more sense. Yay, so it's more consistent with what you see inside your textbook. This is our, it's supposed to be six-sided. I'm sorry about that. That's supposed to be a straight line. Six-sided glucose molecule, and it's going to get oxidized. Remember, we take a 1 6-carbon sugar and cleave it into 2 3-carbon sugars. Anything that we've added electrons to, like we have up here, we have reduced it. Okay, and which means we now increase the potential energy of these molecules, which we take advantage of in a few minutes. Let's get rid of that. Let's get rid of that, and let's go here. So how do we actually make ATP? Your textbook is just really vague. I don't know why they dropped this in here on you. Some of this you should have gotten in biology. Most of you haven't taken a college-level biology class, and those of you who have, some of this is going to sound familiar, where we can actually take an ATP molecule and we're going to directly transfer it to something else. The example I would give you here would be we take a phosphate from creatine phosphate. So let's draw that up here. Remember from muscle back in AMP1, change colors so I don't confuse you guys. Remember we have creatine, which is free floating inside your interstitial fluid inside between your cells and intracellular fluid inside of your cells. We can add a phosphate to it. We make creatine phosphate equals CP. And that is essentially substrate level phosphorylation inside of your muscles. And then what happens is when we burn that ATP at the myosin head, we now have ADP floating around. And then what your body does is it takes that phosphate from creatine phosphate and now attaches it so it's ADP. We're going to add the P. So now we have ATP. Okay? So that would be substrate level phosphorylation. What we're speaking primarily about today, especially where you're paying attention to the real details, is removal of electrons to pass them through the electron transport chain. So everything that we're going to break apart from our glucose and or our triglycerides and or our amino acids can be passed through the electron transport chain to bind ultimately to oxygen. And that's why it's called oxidative phosphorylation. You inhale that oxygen so 2 hydrogen protons can bind to it. Okay? And unless you're a plant, we're not going to worry about containing chlorophyll-containing plant cells. But all those salads you ate, they got all of their energy through photophosphorylation. So let's get us out of here. And let's go down here. That cartoon I drew a moment ago, right? You have the body bringing in all of the materials. The fate of glucose depends on the needs of your body cells. Remember, 20% of everything goes to your kidneys, 20% goes to your central nervous system. That's both glucose and oxygen. But we can make ATP from amino acids, glycogen, and or triglycerides. Okay? So what's going to happen is we're going to do this. We can either make ATP, or if we have enough ATP, we can make ATP from amino acids. We could make it through glycogen, or we could make it through triglycerides. But let's begin our conversation with what happens with glucose, because we've been talking about this since the very first day of class. Right? Remember, you have a cell. You have a cell. And you need to get glucose inside of it. So we have to get glucose inside. That means we need GLUT transporters. Remember, GLUT transporters are regulated by the amount of insulin. And today we throw just one more piece of information on that. And that is insulin actually causes the insertion of more of these transporters. And therefore, now you have a specific mechanism to get glucose in. I'm going to call that 1,6. Where 1,6 today stands for 1,6-carbon sugar. That is glucose. Okay? And remember, it gets trapped inside here because 6 now gets phosphorylated. So that substrate level phosphorylation is used to trap glucose inside of the cell. Look how that 6 just looks a lot like a G. So it makes sense, I hope. So 1 glucose or 1,6-carbon structure is phosphorylated once inside the cell, so it can't get out. So what's going to happen? Well, we have to break apart that glucose once it's actually inside the cell. What we do is we break it apart into 3 major steps, even though there are 4 that are listed for you here, right? First of all, we have to understand what glycolysis is. So let's go with that. Then we're going to take the products of glycolysis and make this. Okay? And the significance there is the acetyl coenzyme A is only made if oxygen is present. The acetyl coenzyme A, if oxygen is present, then moves inside the mitochondria in mitomatrix. Okay? That's inside the mitochondria itself, which will then allow materials to be produced that are reduced, that will then transport to this structure here, where we take oxygen, O2 plus H plus, and we make water, okay, as well as ATP. And you'll notice here, this is why we say it requires oxygen. This is why you breathe oxygen, okay? So let's break this down. Probably the most important figure you actually have inside your textbook, because it strips away all of the sort of very fine granular details and compartmentalizes what's actually going on. So let me translate that for you. When it's 3 o'clock in the morning and you want to make sure you understand where the important steps are happening, this is what we're showing you here, okay? So glycolysis was happening inside cells long before mitochondria were ever inside of your cells. So what's actually happening inside glycolysis? We'll take the word apart. Glucose, lysis. You're cutting glucose. 1 6-carbon sugar is going to be converted into 2 3-carbon structures that will be converted ultimately into this molecule we're going to call pyruvic acid. Notice 1, 2, 3. 1 6-carbon sugar into 2 3-carbon sugars. In the process of doing that inside of the cell, it makes 4 molecules of ATP. They don't give the numbers to you here. Don't really worry about that. But it costs 2 to do this. And in the process, you make this thing that's called NADH plus H. And these are those electrons and hydrogen protons that are going to be collected and sent to the electron transport chain. And you'll notice through multiple stages of this entire pathway, we're going to collect NADH plus H plus and or FADH2. So what's going on? Well, we're segmenting the responsibilities of detecting if oxygen is present or not. Notice the breakdown of glucose gives us pyruvic acid. Glucose to pyruvic acid. Pyruvic acid will be converted into acetyl coenzyme A. That's important. We make acetyl coenzyme A if oxygen is present. In the process of actually making that acetyl coenzyme A, the waste product is carbon dioxide plus NADH plus H plus. It is that acetyl coenzyme A that is permitted into the inner mitochondrial matrix. Okay, so let me draw mitochondria for you here. It is a double membrane bound structure, right? And what I mean by permitted in is if acetyl coenzyme A has been made, it actually is allowed into the middle of the mitochondria here. Hundreds of thousands of them in there. They undergo this Krebs cycle thing. And the breakdown products, the catabolism of acetyl coenzyme A as it passes through the Krebs cycle is to make some number of ATP molecules, some number of carbon dioxide, some number of NADH plus H plus, and FADH2. And notice where all of these lines are going. Okay, they're taking us to this thing that's called the electron transport chain, where what we're going to do is make ATP from that reduced potential energy. And because you actually inhaled oxygen, all these hydrogen protons we saw coming from over here being, look, hydrogen proton there, hydrogen proton there, hydrogen proton here, they're going to mix with that O2 so that we actually make H2O. So how do we do that? Let's get rid of those drawings. Let's move to the next slide. I'm going to show you this, if only just so you understand that we take 1 6-carbon structure, we make 2 3-carbon structures. And now notice how quickly I move along. Okay? Because once we actually have that pyruvic acid, each pyruvic acid is converted into 2 carbon acetyl groups. And you're making this thing right here. It's a waste product. You're literally decarboxylating. Put that in there. Decarboxylation. as a waste product. That's what you're going to exhale. Think about all the time we thought about how carbon dioxide needs to be exhaled. And remember, it's going to come in different forms. Free carbon dioxide, bicarbonate, okay, as well as carbaminohemoglobin. But you still have to exhale it out. Decarboxylation. Here you go. And what's going to happen is, in the process of doing this, each pyruvic acid loses 2 hydrogen atoms. So NAD plus is reduced to NADH plus H plus. And now you actually have an acetyl coenzyme A, which can move into the Krebs cycle. And let's see what that looks like. So you remember what my horrible cartoon looked like a moment ago? Okay. Remember I drew something that looked like this. Oh, here's a mitochondria. Oh. An inner mitochondrial matrix. And remember I said if pyruvic acid was made and there was oxygen present, it moved inside here. Yeah, their cartoon's much prettier than mine. Mitochondria, outer mitochondrial matrix, inner mitochondrial matrix. Some fluid material inside here. You're going to see that's really important in the future, but pyruvic acid comes in. You remove one of those carbon dioxides, take that energy, bind it to NAD, so you have NADH plus H plus. That gives us an acetyl group, which will bind to the coenzyme A. We don't care where it comes from inside this class, but now you've made acetyl coenzyme A, and notice here it says it can enter the Krebs cycle. This is where usually students raise their hand because they took some class that said, well, if I don't have any oxygen, what happens? Well, that's the anaerobic pathway. Okay? If there is no oxygen, you end up being shunted to the anaerobic pathway where you make 2 lactic acid molecules, and you will convert that back to pyruvic acid if oxygen ever becomes available soon enough. Okay? So this is a pretty good place to take a break. It's only 16 minutes in. I didn't want to jump into the Krebs cycle, though, and then have to sort of speed through it. So I'm going to stop here, and I'm going to start another one in a couple of minutes. Okay.